Hello all, this will be our last topic for Unit 2, and as I mentioned on the last video clip here, we've been talking a little more and more and more about proteins and how vital they are for our bodily functions. You heard about several different kinds of proteins when we were learning about tissues in the skin in chapters 4 and 5, and then we just talked about how critical enzymes are, and enzymes are also types of proteins. So we want to learn a little bit more about how proteins are built. And we also want to learn about how the DNA in our cells serves as a set of chemical recipes or chemical blueprints that tell our cells how to build the proteins that they need. All right, so a little bit more about genes. We hear about genes all the time. Genes are, as we talked a little bit about in Unit 1, these are actually locations on DNA molecules. All right, and all DNA molecules they're two strands of A's and G's and C's and T's, and they're bonded together, and then they're twisted so that they have that, what we call a double helix shape. But that's really all they are. They're these long chains of A's and G's and C's and T's. But the order, the sequence of those A's and G's and C's and T's can wind up functioning almost like chemical letters that provide instructions that tell the cell how to make proteins. All right, the proteins in your body lead to what we call our traits. So what's a trait? That's anything you can observe about a living thing like a human. Hair color, eye color, skin tone. Are you more likely to get certain types of cancer than somebody else? Are you more likely to come down with certain types of infections versus somebody else? Those are all our traits, and even though it's kind of abstract to think about, your traits can generally be traced back to one or more proteins that your cells make, because proteins are the workhorse molecules of our cells. And so things that happen in the body can almost always be traced back to one or more proteins. So proteins are very important, which means we need really good instruction manuals that tell our cells how to make those proteins. And when a cell is going to divide and become two cells, it certainly has to pass those genetic recipes on to both daughter cells. Otherwise, the daughter cells won't be able to make their proteins. You can't make your proteins, you're not going to be able to survive. All right, so these protein codes that we have are really important. All right, so inside your cells, each of your cells has 46 chromosomes, and you get one set of those. Well, let me back up here for a second. You actually have two copies of chromosomes 1 through 22. They are actually numbered. And then you have two sex chromosomes, and those are designated X and Y. All right, so you get two copies of chromosomes 1 through 22, and one of those comes from your mom, one comes from your dad when sperm meets egg. So that accounts for 44 of them. If you're a female, you have two, what we call the X chromosomes, your XX. That's because you got an X chromosome from your mom, and you got an X chromosome from the sperm cell from your dad that contributed to making you. If you're a male, you have an X chromosome and a Y chromosome. So you got the X chromosome from mom, the Y chromosome from dad, and that makes you a male. This little diagram you see over here, this is a type of special imaging that can be done with microscopes and computers, where you can actually zoom in and take a look at what individual chromosomes from one of your cells look like. And that's a technique that can be used to check for some types of genetic disorders, genetic problems. Chromosomes, again, are, I'll wipe that out for a second, very long DNA molecules, complexed with proteins so that they can be wadded up and will fit inside the nuclei of your cells. And again, as we've already mentioned, DNA molecules are chains of A's and G's and C's and T's, and along some of those stretches, if this is a DNA molecule right here, along some stretches, like say from right here to right here, and over here maybe from right there to right there, and over here, maybe from right there to right there, you're going to have stretches of A's and G's and C's and T's that function as genes. And genes are recipes that are going to tell the cell how to make some kind of protein. So that's important stuff to be thinking about. What's going on in between those genes? I mean, you still have A's and G's and C's and T's in between, and in some cases we don't know what those sequences are doing. They may not be doing much of anything. Later on in the future, we may find out a whole lot more about what they're doing. In other cases, they're involved in helping determine whether or not certain genetic recipes are going to be read or not. And so it gets a little more complex than what we're going to be able to talk about here. But you do have gaps between your genes on your DNA molecules. All right, here's another look at that here on this particular diagram. They're just showing you here, inside our cells, chromosomes are in the nuclei, and so if you took, that's what a chromosome looks like right before a cell is getting ready to divide. When you see that crisscross like that, that chromosome has been duplicated because that cell is going to divide and the daughter cells are going to get one of those two copies. But then if you took that chromosome, that's when a DNA molecule is super condensed, folded up, wadded up so that it will fit inside a nucleus. If you could zoom in really closely, you would eventually get to that double helix DNA molecule shape that we talk about. And again, you can have particular stretches, like right in here, along a DNA molecule where that AGCT sequence is functioning like a chemical book, a chemical recipe that's going to tell our cell how to make a particular kind of protein. And it's all about the sequence. What's the order of A's and G's and C's and T's that you have there? Those are functioning very much like letters, even though they are chemical molecules themselves. And how this works, it goes through a series of steps, and we're just going to kind of hit the highlights. One of those is called transcription, you see written there, and the other one is called translation. So what transcription is, if you have a gene on your DNA, like let's say it's this one right in here, that gene is on DNA. DNA is in here. It's inside the nucleus of the cell. Do you remember where we said proteins are made in a cell? Remember the ribosomes, those are the little machines that build proteins? Well, they're sitting out in here. They're in the cytoplasm. Many of them are attached to the endoplasmic reticulum, the ER, to build membrane proteins or proteins that a cell will wind up releasing. So we kind of have a dilemma. How in the world are we going to get this instruction manual from in here to out here? Because the ribosomes have to read it. They're going to need to read those instructions. All right, so that's where this process, transcription, comes in. If you have a gene on DNA, this is your gene on DNA, we make an RNA copy of the gene. So it actually gets copied. There's an enzyme that does that. We said enzymes do chemical reactions. So you have an enzyme that will read the DNA, and it's going to make what we call an RNA copy of that. And remember, your RNA has U's in place of T's. So you don't have, wherever you had a T in DNA, you'd have a U in RNA. All right, and then once you make that RNA copy, the ribosome is going to read that RNA copy, and it's going to do translation. And that's a second process by which you go from RNA to protein. Now, what did we say the building blocks of proteins are? Amino acids. What are the building blocks of our DNA and our RNA? Nucleotides. So that last process is called translation because you're going from nucleotide language in your DNA and your RNA to amino acid language in your proteins. All right, why again are proteins important? You know, why do we need these recipes in our DNA specifically for proteins? We've already heard many different examples. Hair and nails, keratin, the keratin coating on your skin, all those protein fibers in your tissues. Many proteins are enzymes. Let's see, hang on here. It's like my little tablet, my little pen's not writing on the tablet. Many are enzymes. Many are involved as cellular construction workers, like when a cell is going to divide into two cells, it takes a whole lot of work to make that happen, so many of them do those kinds of processes. Many of them function as chemical messengers that are sent between your cells. Many are ion channels that are critical for your brain and your nerves and your muscles to function properly. You have a protein in your blood called hemoglobin that is responsible for binding oxygen gas and transporting it throughout your body to make sure that all of the cells in your body have adequate amounts of oxygen gas. So proteins are very diverse, and it all goes back to, all right, you got these 20 amino acids, different kinds of amino acids floating around in your cells, and it's the order that you put those together in that determines how those proteins are going to fold up into their little 3D structures and take on their little special jobs that they're supposed to have. Okay, so I'm sure we've established by now that proteins are rather important. Here's another look at those two processes we were talking about, transcription and translation. Very important. So if you have a gene on DNA, this is supposed to be like a generic gene that we have on here. Now most genes would actually be a longer stretch of A's and G's and C's and T's. Remember, your DNAs are double-stranded. You have A's bonded to T's and G's bonded to C's in your DNA. So you have that first process that's called transcription, and you're going to make a copy of one of those two DNA strands. RNAs are not double-stranded. So transcription, you make the RNA copy. That's called mRNA or messenger RNA. That's what that stands for. So if you look closely, this messenger RNA here is a copy of that one, that strand of DNA. All right? So that happens in here in the nucleus because your DNA is in the nucleus. Then the messenger RNA, let me wipe this out. There's your messenger RNA copy. It has to go out into the cytoplasm, and then here's a ribosome over here. Remember, ribosomes are these little machines that are going to read the messenger RNA. They literally attach to it, and they slide along, and they read that sequence of A's and U's and G's and C's, and they interpret that. That lets them know which amino acids to grab and bond together to build a protein. All right, so that part is called translation. When you are taking amino acids and bonding them together to build a protein, and you're doing that the way that the ribosome knows which ones to bond together is by reading that messenger RNA. That process is called translation. All right, so you have your chain of amino acids. That can be called a polypeptide, if you remember that. And when it folds up into its nice three-dimensional structure, polypeptide sometimes means a shorter chain of amino acids. It can be okay to use those terms a little bit interchangeably. A protein, though, has a nice folded up three-dimensional structure, as we've learned. And then proteins lead to your phenotype. Phenotype, if you've never heard that word before, is just another name for a trait. So proteins lead to some kind of trait that you have. For example, if the protein we're making here is a sucrase enzyme, sucrase enzymes give you the trait of being able to use sucrose as a nutrient. If you didn't have sucrase enzymes in your digestive tract, you would not be able to use table sugar as a type of energy. Probably a lot of us would like that. If that would if that would happen, then we could probably eat a whole lot more sugar and we would never absorb the calories from it. Remember melanin from when we were learning about the skin. Hopefully you do, because that wasn't too long ago that we were discussing melanin. But melanin is not a protein, but the amount of melanin that you have in your skin determines your skin tone, whether you have a lighter or a darker skin tone. But you have genes that code for enzymes that you need to make melanin. So the gene provides the instructions to make protein enzymes, and then those proteins build the melanin for you. All right, if that makes sense. So even though melanin is not a protein, you would not have melanin if you didn't make the proteins that manufacture it. Well, guess what? If you need that protein, like let's say this is your protein here that's going to build melanin for you, you got to have the gene. You've got to have the genetic recipe for that. Melanin is not just going to, or that enzyme is not just going to spontaneously make itself inside your cell. You've got to have the genetic recipe for it so you can do transcription and translation to make that happen. All right, just a little bit about, we're not going to go into the details on this, but when we're saying, okay, you've got this messenger RNA copy of a gene, and then you're going to do this translation process, and we said the ribosome can read this, and that tells it how to link amino acids. These are all amino acid names: methionine, arginine, tyrosine, and leucine. Those are 4 examples of the 20 amino acids that are floating around in your cells that are used to build proteins. So how does that happen? Well, as it turns out, as a ribosome is, I'm going to wipe this out for a second. What the ribosome does is it looks at the A's and the U's and the G's and the C's in sets of 3. Okay? So like right here, when a ribosome sees AUG, it will always insert, I know you guys can't really see this table probably very well, but this is called a codon table. Codons are sets of 3 of these AGCU bases together, and every 3 of those specifies, well, not all of them, but most of them specify some amino acid you're going to use to build a protein. Okay, so for example, AUG specifies that the ribosome is going to put this amino acid, methionine, into the protein it's building, always. And then it slides over and it reads these next 3, CGG. All right, now you can actually look at a table, CGG, on our table there. If you look really closely, this is what it's showing you. Over on the left, you've got, well, which series of 3, which codons specify particular amino acids? All right, and the little sphere there is the amino acid that is specified. So ARG is an abbreviation for an amino acid called arginine. So you'll have an arginine in this protein that we're making. UAC, if we look on our table over here, we can find it. UAC is right there on the table, and that specifies an amino acid called tyrosine. And it really does work this way. It's almost like the ribosome is reading this table. Obviously, you don't really have a table inside your cells, but this works according to these rules, and that's why you can always faithfully go from this AGCT lingo you have on your DNA to this AGCU lingo you have on the RNA copy, and then finally to this amino acid lingo you have in your proteins, because it always follows this set of reading rules. So it really is like your ribosome is reading these chemical letters, and that's telling it which amino acids to grab and link together to build a protein. That will happen until you hit what's called a stop codon. So eventually you got to stop building the protein. So there are some series of A's, UAA, UAG, and UGA, those 3. When the ribosome sees that and reads that, it quits. It stops building the protein. All right, and don't worry about the nitty-gritty details there. No, I'm not going to ask you to do this, or to read a table like this on the test, although it's really not that hard if you have to. And actually they do put that on the TEAS test. If you haven't taken the TEAS test for entry into nursing school, they will have some of that information on there. Take microbiology, you'll get quite a bit more of this as you go through the genetics part of that particular class. All right, so again, relating this back to a gene on DNA. All right, back over here. You got to have the proper code on your DNA in order to have the proper sequence of amino acids in the protein that gene specifies, that gene is a recipe for. So if this code over here gets screwed up, that's called a mutation. Like if you had an A up there and it got changed over to a G, that's called a mutation. And if you screw this code up, you may wind up screwing up your amino acid code down there. You got to have the right sequence of amino acids. This is an example of an amino acid. Here's your chain of amino acids linked together to build a protein. That's called your amino acid sequence. And that determines how a protein folds into its 3D shape. If you screw up the amino acid sequence, you have a different combination of those 20 amino acids in there, your protein is going to be something completely different. It's not the same protein anymore, and so it's not going to function like it's supposed to. So thinking back, this is why when a cell divides, it really doesn't need to already have all of the proteins that the daughter cells will need. It just needs to pass along these genetic recipe books. It's kind of like, if you had two kitchens and you wanted to make the same kinds of foods in both kitchens, you wouldn't automatically need all of those foods present in both kitchens. As long as you pass along the recipe book from one kitchen to another, you're going to be able to duplicate what you're making in that second kitchen versus the first kitchen. It's the same thing with cells. Cells need many different kinds of proteins to be able to function properly, and as long as a cell, when it divides, it copies the DNA and passes those recipes along to both daughter cells will have the capability of making all the proteins that they need, and so they should have the same traits that the mother cell had. So that's why DNA a lot of times is referred to as your inheritable or inherited molecule. All right, it all goes back to our DNA as we've heard before. All right, one last little thing here. The Human Genome Project, you might have heard of that before. It's been in the news. This term genome, the human genome, that's just referring to one set of human chromosomes. Okay, so the set of chromosomes I have in the nuclei of my cells that's my genome. Your genome is the set of chromosomes that you have in your cells. Now we all have two copies of 1 through 22, and then you've got your two sex chromosomes. But there are some little quirks. There are some differences between my genome and yours, as we'll see in a second. The Human Genome Project, which was finished 10 years ago in 2003, the goal there was to sequence completely the human genome, determine the AGCT letter code for all of those chromosomes. And so that was completed in 2003. The human genome, a whole human genome, that's just one set of chromosomes, consists of 3 billion base pairs. A base pair is an A bonded to a T and a G bonded to a C on a double-stranded DNA molecule. So that's how the lengths are reported in base pairs. About 3 billion base pairs in a human genome. My genome is about 99.9% identical to yours. So between human beings, just 0.1% differs. That means in some locations where I have an A, you might have a G, or where I have a C, you might have a T. So, but at just 0.1% of locations will we have differences on average. But nevertheless, those differences are what give each of us our own unique traits that we have. Our appearance, our personalities, things like that. Anything that you can trace to genetics. As far as we know, so along this 3 billion base pairs that you have divided up into these 23 chromosomes, we know that there are about 20,000 different genes that code for proteins. So, and then those proteins can get put together in different ways. So we make even more proteins than that, but 20,000 different genes along those chromosomes that can be used as recipes for building proteins. As part of the Human Genome Project, now we have all this information. Now they're trying to figure out, well, that's nice, we've got all these genes. What are those genes doing? What do they code for? In a lot of cases, we don't know what they code for. And what are those proteins doing? The recipes for these proteins, what are the functions of the proteins? Are those proteins important in some way for human health issues like cancer or various genetic disorders? In some cases, we know which proteins are playing roles in those, and in other cases, we do not. Okay, so then we have a really hot area of biology and science today called biotechnology, where you're using this information from biology to try to further the human condition. And so in biotechnology, you do things like figure out what the functions are of particular proteins, identifying whether certain diseases are caused by genetic mutations. Many diseases do have a genetic basis. And, can we replace defective genes in the future? Can we, let's say somebody has a disease like cystic fibrosis, which we do know which gene is involved and is screwed up in people who have cystic fibrosis. So maybe sometime in the future, not too far down the road, we'll actually be able to replace those defective genes with good copies in people who have those types of problems. So it's a really exciting area, and it's going to really change the way it's going to change human health. It's going to play an important role during your careers going into the health sciences, because there are going to be a lot of new medicines that are based upon this type of information. Probably not too far down the road, you will be giving your patients medicines to correct genetic defects. Those are things that we really could have only dreamed about doing just a couple of decades ago. And I think during your careers, you're going to see those types of things happen. All right, just want to talk a little bit to you about genetics here. Certainly just hitting some highlights. It's also a very complex topic. You'll learn more about it when you take microbiology for sure. It will come up again occasionally during your anatomy and physiology courses. And I also think it is a very important topic in 2013. You're moving forward and going into the health sciences career. Medicine is going to change, and a lot of new medications are going to have this genetic type basis. And so I do want you guys to have at least a little bit of a surface understanding of what that's all about. All right, so that concludes, that actually concludes Unit 2. So you are ready to keep working on studying and reviewing and practicing, and then going on to take your exam. So good luck with that, and please let me know if you have questions about any of this. I'm more than happy to answer those. And as I've mentioned before, I would love it if you guys would pose questions on the discussion boards. That way other students who are having the same questions and maybe too bashful to pose them can benefit from those answers as well. Your questions are not stupid. I know there's a lot of information here that is new to you if you've been out of school for a long time, or even if you just got out of school. Sometimes you don't have a strong background in science, and you can get kind of waylaid here on the front end of a class like this with all the new terminology and concepts you have to learn. So please come to me for help, and use the discussion boards as needed. No question is a silly question. If you don't understand something, I can help you clear it up. All right, thanks a lot, and good luck with your preparation for exams.